right, introduction to genetics. We're just gonna do a brief unit. It's only gonna be like a week and a half, so 4 days, so I guess 2.5 weeks, because I only see you guys 2 days every week. Okay, so we have this guy, Gregor Mendel. He was an Austrian monk in the mid-1800s. He is considered to be the father of genetics. And genetics is the science of heredity, or how things are inherited. So we talked about last time meiosis and how things are inherited from your parents and how they get passed on. And now we're going to actually talk about what is in those chromosomes and on the DNA. So Gregor Mendel also developed the laws of heredity. And like I said, heredity is the passing on of traits to the next generation. So how he did this is he studied pea plants. So he was a monk. He didn't have amazing things to look at. So he grew and studied pea plants. And he followed various traits in the pea plants that he bred. So things like the flower color, where the flower grows, if it's on the bottom or if it's on the top, seed color, seed shape, pod shape, and pod color, and also stem length. So he studied most of those traits. Then in 1866, he published his findings on the methods of mathematics of inherited traits in his pea plants. Like I said, he studied 7 different traits, and I read all those. It's in the diagram on the picture also. It's also in your notes, if I can find them ever. All right, so what did he notice? He figured out that some pea plants produced green seeds and some had yellow seeds. So he looked at the color, like I said. And in order to understand how these traits were inherited, he cross-pollinated the pea plants by transferring the male gametes. So remember the sperm, or the sperm-like male, I think it's called the stamen, in flowers. And so he transferred that from a purebred green-seeded plant to a female reproductive parts of a purebred yellow-seeded plant. So he took one that he knew would always be green, and he took one that he knew would always be yellow, and he cross-pollinated them to see what he would get. So when he looked at this, he was looking at seed color. And there are 2 forms of the trait, yellow or green. One was dominant and one was masked or hidden. And remember from our dragons that we studied, dominant just means the one that shows up or is observed or seen. So overall, he concluded that individual factors must control the inheritance in pea plants. So the seed color is different than seed shape. So they are inherited separately. And that the traits must exist in pairs in the female, and the female parent contributes one factor and the male contributes the other. So just like we found with our chromosomes, one chromosome comes from mom, one chromosome comes from dad, or like the male and the female chromosomes. I'm going to try and assign this one more time. So by studying things like pea plants and mice and flies, it's a lot easier to go through traits because they reproduce a lot faster than like humans or elephants or mammals, larger mammals anyway. So this was kind of his, experiment that he looked at. So there was a parental generation. We just call it the P generation, like the letter P. That's the parent generation at the top. And then the second generation after you cross the parents is called the first filial generation. You don't have to memorize that or anything, but it's F1. So if you look right here, the parents are at the top, and then the first filial F1 is right there. And that's the offspring from the parents. And then they took all of these pea plants from the F1 and combined them together to get the F2. So the second filial generation is taking all of the offspring from the parents and combining them together, or cross-pollinating them. And that's when he got a bunch of different colors. And we'll see this ratio, the 3-to-1 ratio, we'll see this in what we're going to do if we get to it today. So what he saw in terms of words and visually what he saw was that the purebred yellow-seeded plant was cross-pollinated with a purebred green-seeded plant. And remember, purebred just means they're always green or they're always yellow. And then in the F1 generation, so the first offspring generation, he saw that they were all yellow, and then he cross-pollinated all the yellow ones together, and he saw that they got a ratio of 3-to-1 yellow to green. So mostly yellow, some green. All right, so just some vocabulary that you're going to need to know or be familiar with that I'll say, and then if this ever assigns, you'll be able to write it down. But the factors that control your traits are your genes. We kind of already talked about that. Genes are on chromosomes. An allele is an alternative form of a single gene passed down from generation to generation. So it's just one of the types of the genes. Alleles can be represented by letters. So how we write them down scientifically when we're trying to figure out who passed on what, we just give it a letter. So for example, if we were doing this with the seeds, capital Y would be a yellow seed and a lowercase y would be a green seed. Genotypes are just represented by two alleles. Geno meaning genes, so it's just the letter representation. So two uppercase Y's, an uppercase and a lowercase, or two lowercase, which we'll learn what that means hopefully today if Google Classroom decides to. Just some more, vocabulary. There's two types of genotypes. There's two types of those genes. There's homozygous and heterozygous. Homo, the prefix in Latin, means same. Homo sapien, homozygous, same. And the example would be two of the same letters, two uppercase Y's, two lowercase T's, two uppercase B's, two of anything the same. Homozygous genotypes mean the alleles will be the same, like I said. They will either be two dominant, so two uppercase, or two recessive, which are two lowercase. We'll review those terms in a little bit. So when you put two dominant alleles together, the trait is always dominant because the uppercase one means dominant. It's the one that shows up, and it doesn't have an option, so it's going to show up. When you put two recessive, or the little letters together, that trait is going to be recessive, and it's also going to show up, though, because both of them say recessive, so the only option to show up is recessive or the little R. I don't know why I can't see the bottom one. But homozygous organisms, either homozygous dominant or homozygous recessive, are called purebred. So that was the yellow and the green. All right. Heterozygous means two different. Hetero, the prefix, means different. So that would be one uppercase letter and one lowercase letter when we're writing it out scientifically. What else? So they being different. There will be a dominant allele, so the uppercase, and then there will be one lowercase. That always means heterozygous. So if you just say the person was heterozygous for dimples, that means they have one uppercase allele and one lowercase allele. So one of each. What else? And when we use the term hybrid, that means one of each. You can think of it like we're doing hybrid learning. There's some people remote, there's some people in person. Hybrid means one of each, so both. All right, and then another word is phenotype. So we just talked about genotypes, it's the letter. I think of it as like a code. And then phenotypes are the physical or observed characteristics. It's the physical appearance of the trait, such as seed color, hair color, or eye color. So it's not saying the alleles, it's not saying uppercase Y, uppercase B, lowercase b. It's saying brown hair, blue eyes, yellow seeds, green seeds. So it's the actual trait, like the description word. This is our last slide, so I'm going to see what happens today. But the law of segregation is kind of what we learned about in meiosis. It's that the two alleles, so the two letters or the two chromosomes for each trait separate in meiosis. So remember when the gametes separated from mom and dad when they got the sperm and egg cells, how they separated? That's the law of segregation. And during fertilization, when the sperm and egg combine, the two alleles for that trait come together, and that's how you determine which one's dominant and which one would be recessive. So this is just a diagram kind of explaining it. If you want more detail, I'm not going to ask you about this diagram. We learned about meiosis, how they separate, and now fertilization is taking them together, and we're learning which one shows up and which characteristics are dominant or recessive. Oh, we do have another one. So we're going to get into Punnett squares, maybe today, maybe tomorrow. I'm trying to assign this to you still, but Google Classroom is not cooperating. But a Punnett square is basically just a device or a square. You guys might have used it before. That shows, or it's used to predict or figure out the possible offspring of a cross between two known genotypes. So if we know the parents, like letters, if we know their alleles, so in here we know that the yellow one was two uppercase Y's and the green one was two lowercase Y's. we could make a Punnett square to figure out what all of the offspring would look like. So like I said, Punnett squares are used to determine probability of getting a certain trait. The results are predicted by the law of probability and are more likely to be seen when there is a large number. So if you only take like 4 of the offspring, it's probably not going to show up the same way as if you had 100 pea plant offspring. And then just another term, monohybrid, mono meaning one, is a single trait that we are looking at. So looking at seed color or looking at seed size. Instead, a dihybrid would be looking at both of them together. But all we're going to work on is monohybrid, where it's single traits. So this is a Punnett square. I'll try and explain it to you without, actually being able to look at it. But so the parents are put on the outside of the square. So they put the mom over here on the top, and then the dad, I think, over here on the side. So they each have 2 alleles. The dad has or both of them, the mom and the dad had an uppercase T and a lowercase t, and you put them on the side. So you just separate them into the boxes. So the uppercase T went to the top box and the lowercase t went to the bottom box. A rule when we do Punnett squares is that the uppercase letter should always be written first. So then you just kind of take them and you bring all the ones on the top down. So if this one had an uppercase T, you bring it down to all these squares. So there's an uppercase T from the top, and then an uppercase T from the top here. And then you take the little t and bring it down. The little t over here and the little t over here. And you do the same thing across. We're going to practice this if Google Classroom would ever work, but I'm just showing you how it works right now. So the uppercase T on the side gets put across, and then the lowercase t on the side gets put across also. So that's what you technically should be working on. I'm going to see if Google Classroom will ever decide to work. I'll stop recording the notes section for right now.